Well, good afternoon and welcome to SciFest Africa, South Africa's national science festival and a project of the Grahamstown Foundation. So SciFest Africa 2021 is celebrating 25 years in science engagement. SciFest Africa was established in 1996 with an intention to promote the public awareness, appreciation, and understanding of science, technology, and innovation. Since 1996, SciFest Africa has been educating and enlightening its audiences with a feast of discovery, offering a crucial doorway into the world of science, technology, engineering, mathematics, and innovation for people of all ages. The 2021 theme, Creative Science, is inspired and aligned to the International Year of Creative Economy for Sustainable Development, as proclaimed by the United Nations. So my name is Michelle Ramsey, and I am a geneticist from the University of the Witwatersrand, and I will act as the host this afternoon. So it gives me great pleasure to welcome our speaker, Dr. Megan Hochstrasser from the Innovative Genomics Institute at the University of California in Berkeley. And she's going to talk to us about CRISPR and rewriting DNA and the future of humanity. So just to tell you a little bit about Megan. So Megan has a BA in biology from Brown University and received her PhD from Jennifer Doudna's lab at UC Berkeley in 2016, where she studied mechanisms of CRISPR immunity in bacteria. Now, for those of you who are not aware, Dr. Doudna was the joint recipient of the 2020 Nobel Prize in Chemistry for the discovery of the CRISPR technology. But back to Megan. So Megan joined the Innovative Genomics Institute in September 2016, hoping to bridge the gap between researchers and the public. Megan develops educational resources and programs, spearheading everything from mobile app development to community event planning. She regularly gives talks to students, teachers, scientists, journalists, physicians, and more. And as a CRISPR expert and professional science communicator, Megan brings diverse audience into the conversation about genome editing. We're absolutely delighted to welcome Megan to give her presentation and looking forward to a Q&A session at the end. And just to ask everybody, please, to put your questions in the Q&A box where we can look at them and take them up at the end. So her talk will be about 30 to 40 minutes. So we'll have a good opportunity to explore some of the questions that you may have. So Megan, take it away. We're looking forward to your talk. Thank you.
Thanks so much, Michelle, for that nice introduction. And hi, everyone. Great to have you here today. Let me just get my screen up. All right. So as Michelle said, today I'm going to be talking to you about CRISPR. My name is Megan Hochstrasser, and I work at a group called the Innovative Genomics Institute, or IGI, which is easier to say. And we are an institute that is aiming to use genome editing and other genome engineering tools to benefit humanity and make sure that these benefits are accessible to everyone. So our focus is really on doing all kinds of research using CRISPR and other tools, but then also making sure that they get translated back to benefit society. We're a partnership between UC Berkeley and UC San Francisco, so we kind of sound like a company, but we're a nonprofit that sort of floats above these two universities. And my role is in education and outreach. So I give a lot of talks, I make tools, all of the events that you see on the screen were things that I organize. So my mission is always to give people access to information and get people excited about science and science careers, if that's what makes sense for them, and to mostly empower people with knowledge. So I think knowledge is power, and I think of my job as sort of translating complex science so people can understand it and use that information to think about their everyday lives. Okay, so into the talk. If we were here in person, I would say have you shout out if you know what I'm showing you on the screen, but I'll just tell you this is DNA. So this is an artist's depiction of DNA. And everything I'm going to be talking about today sort of focuses and swirls around DNA, because DNA serves as a blueprint for how our cells work, how our bodies work, how any living thing functions. And so if we're able to change that, potentially, we can change how living things function. And that's what I'm going to be talking to you about today. So just to briefly go into what DNA is and why it matters, what it does. DNA, as I said, is a blueprint that controls traits. So traits are characteristics about living things. And I just like to give some examples that I've sort of stolen from 23andMe, which is this consumer DNA testing company. You might have used one of their kits or heard of them. And these are some of the traits that 23andMe can tell you you have or don't have, which you probably already know, but they can tell you why you have these, sort of the genetic basis for why these traits happen for you. So this can include everything from having dimples in your cheeks to having flat feet. And how accurate this is sort of variable for different traits. Some of these are really easy for us to predict based on DNA, and some traits, it's kind of like, you know, you have a 20% chance of having this trait. We're not totally sure always how a DNA sequence relates to a trait. This is sort of fun and frivolous, but there's also a darker side to traits and DNA, which is that you can have mutations in your DNA that cause various conditions with differing degrees of severity. So I've again taken little snippets from what 23andMe can test for. And on the right, I'm showing you genetic diseases. So these are all conditions caused by variations or mutations in DNA. And so some of these you might have heard of, like cystic fibrosis is one of the more well-known genetic diseases. And then some of them have very long names, they're very specific. And so the number of people across the world that are afflicted with these conditions can vary a lot. So there can be, you know, 100,000 people with one condition, there might be 12 people on Earth that have a particular mutation that causes a disease. So this has implications for who's going to have therapies developed for them, which we can talk about a little bit later. But I think the point I want to make here is that for a long time, these traits were just things you were born with. And even if you had something go wrong and you had something like cystic fibrosis, there wasn't really that much we could do about it for a very long time. It's, you know, it's written into your DNA. That's an unchangeable thing for the most part. But that's why CRISPR is so exciting, because we finally have a chance to potentially correct some of these mutations, because CRISPR lets us change DNA. So what I'm showing you here is another artist's depiction of what CRISPR might look like. So this is based on some real science, but there's always some artistic flair. So I'm showing you here, this green blob is called Cas9. That's just one CRISPR protein, CRISPR-associated protein 9, the one that people use the most. And this is a kind of microscopic view zoomed into a cell of this Cas9 or CRISPR protein in action, finding a site in DNA and making a cut. So I'll talk to you about how making a cut in DNA can change its sequence. But I just wanted to show you here that this Cas9 protein is guided by this little piece of RNA, which people are becoming more familiar with because of the COVID vaccines. But RNA is sort of just like a single stranded piece of DNA. So instead of it's like one half of that twisty ladder that makes up DNA. And we as scientists can change this sequence of this RNA, and that tells Cas9 to go to a different place in the DNA. So it's kind of like a little programmable cut machine. So I'll get into that in a little bit more depth in a bit. But I just wanted to show you that, as I said, this was sort of based on some science, this picture. And there's this really cool video. I just like to show this because I think it's amazing, where people use something called atomic force microscopy to actually visualize this incredibly tiny protein directly or indirectly, but it looks like we're looking at it. So let me just play this video really quick. So what we're watching right now is Cas9 sitting on DNA, and it's about to do what it does to DNA. There you go. So you could actually see it cut the DNA. So this was like mind-boggling to me when I first saw it. It's most of science, when you're doing it in the lab, is like moving little bits of clear liquid around and imagining what's in them. So getting to actually see this, I thought was incredible. Okay, so it really does look like that. That's how it works. Let me go into a little bit more depth about how we program it. So if I wanted to edit cystic fibrosis, say I wanted to develop a therapy to treat that disease, I would first have to know the target I'm looking for. So a lot of people ask me about different conditions and symptoms that they suffer from, and CRISPR is really not going to be very helpful unless we know exactly what DNA change to make to try to treat the disease. So with cystic fibrosis, it's usually pretty clear. We'll know exactly where a mutation is. So once we identify our target sequence, then we go about programming the Cas9 protein to go find it and change it. So what this looks like, as I said before, is this, we reprogram this location code in something called the guide RNA. So the guide is sort of half handle. So this one half that the Cas9 protein hangs on to and never changes. But then the other half is something that we change. So we can make this little stretch of 20 genetic letters be anything we want. So we can make Cas9 go target that cystic fibrosis mutation. We can make it go target an eye color variant if we wanted to. There's anything you want to do, basically you can, which brings up a lot of ethical choices. So we'll talk about that later as well. So once we've programmed Cas9 to go find its target, what it does is it goes and it unwinds the DNA and matches it to its RNA. So instead of having this double helix of DNA, it gets unwound and you get a double helix of RNA plus DNA. And this is just sort of the protein's check, like, yes, I've found my target, all of my letters match, that means I'm going to make a cut. So once Cas9 makes that cut, you might wonder, why would you want to cut DNA? Especially if you have a mutation, it seems like damaging DNA is going to be worse than leaving it alone, which could be true. But for the most part, this is how most genome editing tools work. So they all begin with this one cut that initiates additional processes that are what actually changes the sequence. So let's go through this in a little bit. So we can use our molecular scissors, Cas9, CRISPR, other tools to find a target, make a cut. This is also called a double strand break because it cuts both strands of the DNA. And what happens when this cut goes into DNA is that the cell basically freaks out. So our cells have all these systems in place to find damage in DNA and fix it, because damage to DNA can be lethal for the cell. So we need to have these emergency repair systems in place. And our DNA is getting damaged all the time. There's UV light, there's things happening in the cells, there's mutations that happen when it gets copied. So there's all these systems in place to be able to look for and repair those breaks. So this sends the cell into freak out mode and sends some repair proteins to fix it. And so there's two main ways that this can get resolved. And these are important because they have implications for what we can do to change DNA and what kinds of changes we can make. So the first way that this gets repaired is sort of I think of it as like a desperate repair, where the cell just kind of slaps the two ends of the DNA back together. Sometimes it ends up throwing in extra DNA letters. Sometimes it ends up cutting a piece off to make it work. And so it's sort of a sloppy repair that ends up messing up a gene. So imagine you have a gene there and you just screwed up the middle of it. It's like if a gene is a cookbook for a protein, if you just ripped pages out of the middle of a cookbook, out of your recipe, and then tried to make the recipe, it would probably not turn out very well. So the same thing happens with a gene, where it basically is broken by this process. And again, that might sound not useful and bad if you're trying to fix a mutation and treat a disease, but this is actually a really useful outcome in some applications. So like in basic research, when you're just trying to understand how genes relate to traits and how they cause traits, it can be useful to break something to try to get insight into what it did. So for example, there's people that are studying butterfly wing patterning and how the DNA of a butterfly controls what those really beautiful patterns look like. And so they can use knockouts, that's what this is called, where you knock out a gene and stop it from working, to then look and see, okay, what happened in the butterfly's wing? Oh, we no longer have these black lines. So that gene must be in some way controlling those black lines that develop on the wing, things like that. But then, as I said, if we want to treat a disease, we mostly want to have a different outcome. So the other way that this big cut can get repaired is if we, as the experimenter or scientist, add in a piece of donor DNA. So if we want a certain sequence to end up at that site, we have to add that sequence ourselves. So we physically add a piece of DNA along with the CRISPR machinery, and the cell basically gets tricked into pasting this DNA over the cut. So we end up replacing what was there or adding something new. So this is useful if you're trying to fix a mutation that causes disease. It can be useful if you want to add a new function, like if you're studying a plant or something like that, and you want to see what happens if I add this enzyme. Can it do something new and useful? So these are the two ways that the cut can be repaired. I think people often sort of think CRISPR itself is making direct changes, but we don't have that level of control, at least not yet. So it can sometimes be hard to get a cell to do the repair that we want to do. And that's one of the things that I think slows down some of the developments of applications that we'd really like to bring to the world, because we're still sort of getting a handle on how to control this process. Okay, so I know that was a lot of like molecular detail. So I'm going to show you a video to summarize, and then we'll kind of pause before getting into all the things you can do with CRISPR. Okay, so I'm just going to show you a video of all of this happening. So we're going to dive into a cell here, and we're going to go into the nucleus, which is where all the DNA is stored. And we're going to see a bunch of Cas9 proteins searching for their targets. So these are not finding their targets, but the next one will. So this Cas9 is going to search around the DNA, and I won't get into too much how it does this, but it opens up the two strands of the DNA, and its RNA wraps around and makes a little helix with that DNA. And it says, yes, this is my target. I found it, so I'm going to make a cut. So it cuts through both strands. Other proteins are going to bump it off. This is the repair protein that's going to fix that cut. And as you see in pink, it left a mutation. So it added these two nucleotides at that site. And so this is probably going to be enough to knock out that gene and stop it from functioning. And the other process, like I said, I'm not showing here, but in that process, we could add DNA or replace DNA instead of relying on sort of a almost random change that messes things up. So that's the process. That's what we call genome editing. And this is the point where I like to pause. So some people really like understanding the nitty gritty of how this works. Other people are like, okay, it can change DNA. I get it. Let's talk about it. So I like to pause here and just remind everyone that the point here is that we can change DNA. So for example, if we were going to pretend this sentence is the sequence of a gene, and we have an error in what we're trying to say. So it says battle star instead of little star. If we want to change that, we can potentially use a tool like CRISPR to correct the sequence and fix it to be what we want. And so some people think of this almost like text editing. So imagine you have a Word document or a Google Doc or some kind of text editing document in front of you on your computer, and you're just typing to make a change. That's one analogy. So that's why we call it genome editing, which works for some people. Other people like to think of it more as like a genetic surgery, where you're cutting out little pieces of DNA, replacing them with others. And I think all of these analogies and metaphors can work. It depends on how you like to think about it. So I try to present multiple. But the thing to remember from this point forward is that CRISPR lets us change DNA in living cells. Okay, so why is this such a big deal? There have been other genome editing tools. We've had similar types of ability in the past, but everyone went kind of wild when CRISPR came out and scientists got very excited. So why is that? I won't get into this in too much detail, so don't worry, but prior tools, which you may or may not have heard of, they're called zinc fingers and talons. They relied on engineering a new protein, that's what's shown in orange here, every time you wanted to cut at a new place. And so protein engineering is a whole field in and of itself, which made making new edits really difficult. But with CRISPR, because you can just change an RNA instead of a protein, it's much, much easier. So RNA is something that you can just order online. You can order DNA to make protein like I'm showing you here, or you can directly order a new RNA. So this means when you're a scientist in a lab, you might be starting a new project. Within like a week or two, you could get all the material you need to make a genome edit and start making it. Whereas with these other tools, it was like months of engineering and it took a lot more work and it was a lot more costly. So this is a cheaper way of doing it, and it's just much more user-friendly. So people use CRISPR all around the world all the time now. It's become a very standard tool. Okay, so who cares? Why do we want a tool for this? What do we want to do with it? What does this let us do? Well, if it has DNA, we can edit it. And so every living thing has DNA. That's part of the definition of life. And what I'm showing you on this slide is a small selection of the diversity of life. So these are what we would call model organisms, meaning they've been studied in labs for many years. They're kind of the workhorses of biology, where people have all different tools to work with them. And they've taught us a lot about how biology works. And so this is things like fruit flies, mice, zebrafish, yeast, Arabidopsis, that's the model plant. And these are really powerful tools, but they're very limited, right? That this is not the whole expanse of what living things look like. And so with CRISPR, we've actually opened the door to being able to study a huge number of organisms. So CRISPR so far has worked in essentially any organism it's been tried in. And everything I'm showing you on this slide has been edited with CRISPR for one reason or another. And actually, this is a very old slide at this point. So hundreds more have been edited since I originally made this. So this means we can now study cool things like I was telling you about before, like butterfly wing patterns, and apples, we can edit apples, flowers, tomatoes, all different kinds of microbes and molds, mushrooms, ferrets, anything you want. So I'm really excited about the just simple basic research we can do using this tool to better understand nature. Back to some of the other applications that I think people get the most excited about, that would be clinical applications or editing to treat genetic disease. So one of the most prominent examples of this is editing people to treat sickle cell disease. So this is something that's, I think, one of the applications that is the furthest along in development. So there are already clinical trials, multiple clinical trials actually, testing whether or not CRISPR is a legitimate, safe, and effective way to treat this disease. So sickle cell, if you're not familiar with it, involves red blood cells, which are normally round, being turned into this sort of half-moon sickled shape. So the reason that this happens in people, and sickled cells get stuck in blood vessels. So because they're not smooth, they don't travel through the vessel as easily, and they can pile up like you see here and get stuck and block blood flow. So this causes really excruciating pain. And when oxygen can't get to the tissue or organ, it can cause damage. So it's a really, really tough disease to deal with. And it's all caused by this single mutation. So there's one genetic letter in people who have sickle cell that is not present in other people. So this T here is normally an A in everyone else. And this is a mutation in a gene called hemoglobin that carries oxygen, and it ends up making the hemoglobin stick together in these big fibers, which they shouldn't be in, and the fibers bend the cells. So that's the kind of molecular basis for how the disease happens. But because it's just one mutation out of the millions of other letters, it's actually pretty promising as a target for something like CRISPR. So we can, in theory, make a cut around this site, add in a piece of that donor DNA, like I was talking about earlier, and replace this single letter with just the healthy version. So that would be an A in this case. And this is very exciting. This is something that we're working on at our institute using this method where we directly try to correct that mutation. But it's kind of lucky that many, many people are working on this and pursuing complementary strategies. So I think we're going to have multiple approaches being tested, and we'll figure out which is actually best for patients. And this is, as I said, the kind of furthest along that we have. And there's this really incredible example of a woman named Victoria Gray, who I'm showing you here, who's decided to make her story public, which she did not have to do. She could have remained anonymous, but she wanted to show people what was going on and talk about how this treatment, this experimental treatment, is affecting her life. She is someone who's participating in a clinical trial, which is an initial test of a medicine where we're only testing a handful of people, because if you have something that it doesn't end up being safe or effective, you don't want to give risk to lots of people. You want to make sure that only a handful of people who have been thoroughly informed about the risks are able to participate. And so in this early study that tested just a handful of people, Victoria Gray received a CRISPR therapy for her sickle cell disease, and she seems to be functionally cured. And I put cured in quotes just because I'm very hesitant to ever say that for multiple reasons. So one, any existing damage that her disease caused is not going to be reversed, but this could be preventing future damage from sickle cell. And two, we really have to follow anyone who's in one of these clinical trials in the long term to make sure that this therapy lasts, that the effects are permanent, because in theory, this is a one-time correction, and it should last forever. But that's not necessarily true. It's something we have to test in people to find out. And we also need to look for any kind of side effects that we can't see early on. And so this is, people will be following up with Victoria Gray for hopefully, a decade, and hopefully we'll have updates. But so far, it seems like she has lost all the symptoms of sickle cell. So she doesn't need blood transfusions anymore. She hasn't had pain crises, and it's totally changed her life. So this was a really kind of inspirational and amazing story. And there's a lot of other stuff going on as well that I can't summarize here. But I do recommend this post that we put together on our website, where we've listed, I think we've summarized every single CRISPR clinical trial that's happening right now. So there are trials for other genetic diseases that have had promising results. Some are kind of less exciting, but so far there haven't been any huge safety red flags, which is what I'm looking for. So it seems like so far things have been going well, and I'm very excited about what these therapies can mean for people going forward. So this is a great article if you want more information on that. All right, so there are a lot of different applications of CRISPR. I've only talked about basic research and medical applications, but there's so much more than this that I can't get into. And I think this is where I like to sort of talk about the choices that we have as scientists and that you have as people of the world, citizens of the world, in making decisions about how we use technology. So we can edit DNA and we can edit all kinds of organisms for all kinds of reasons, but what should we be doing? What are the best applications? What's the right thing to do? How should we be spending our time and resources? Well, there's a lot we can do. So my slides are very slow today. I don't know why. So I'm going to just show you a couple of examples and then we'll get into one big one. So I didn't talk here at all about editing food and plants for agriculture, but that's another thing that our institute does and a lot of people are working on. And this kind of brings up a lot of issues for some people because, to me, it's great. We can potentially make crops that are resistant to climate change. We can make food that's more nutritious. I think there's a lot of potential there, but some people just feel, leery or icky about having their food manipulated in this way. There's a lot of controversy about prior technologies like GMO technology, you've probably heard of, which is a distinct and different technology that does something different. But CRISPR is, of course, also a genetic engineering tool. They both fall under that bucket. So I think we have a lot to do in communicating about what CRISPR is and what it can potentially do in agriculture. But I think there's also very legitimate objections in some cases to certain applications in agriculture as well. So I think CRISPR is safe on that front. I think that's generally agreed among scientists. But how that's brought out to the world might not be, what's best for the world. I think we all have to kind of make decisions together about what we want to spend time on and who gets to receive things like edited crops, what farmers are being left behind, can everyone access these benefits? Are companies, huge companies controlling the licensing, etc. I won't get into it that much. Another kind of exciting application for gene editing is a concept called a gene drive, which I definitely don't have time to explain. It's pretty complicated, but it's basically something that we can use in multiple organisms, but especially in mosquitoes. So that's what I'm showing you here. And it's something that lets us control inheritance. So we could potentially force a gene through a population or even eliminate a population of mosquitoes. So most people are talking about this because it has a potential to eradicate malaria or attempt to do that, because we could wipe out all of the mosquitoes that carry malaria, or we could attempt to do a more refined approach, which would be to make mosquitoes that can't carry malaria and force that genetic profile through the whole population very quickly. And so this is an interesting question, I think, ethically, because there could be huge consequences to taking an entire species out of an ecosystem, especially when mosquitoes don't honor borders between countries. You know, if you release a genetically engineered mosquito in one country, it can fly over the border and no one will know. And so there could be consequences to doing that are very hard to predict. But on the other hand, malaria kills a horrifying number of people and is a huge problem. And so, you know, someone else might argue we need every tool at our disposal. So there's a lot of really interesting questions to think about there. And I don't think we're quite at the point where we've solved that yet, but I would say as an update of where things are, I think people are generally sort of, if somebody wants to release one of these mosquitoes, they're coming to the committee where they want to release it and talking to them directly and sort of trying to make decisions on this community level. And I've heard of several people focusing on islands because the mosquitoes can't get out for the most part or are less likely to escape, and trying to make decisions there. So I don't believe any gene-drive mosquitoes, CRISPR gene-drive mosquitoes are in the wild right now. That's my understanding, but that could be happening in the near future. Okay, so those are some sort of ethically thorny application areas, but the one that has really garnered the most attention and debate is probably the prospect of using something like CRISPR to edit the human germline. So that means sperm, eggs, or embryo. So these are edits that get passed down to a baby and then their descendants as well. So let's get into that in a little bit more depth. So human germline editing, as I said, it involves editing sperm, eggs, or an embryo. So mostly it means an embryo. And it all kind of builds on IVF technologies. That's in vitro fertilization, and that's what I'm showing you here. So what you're seeing is a human egg cell being injected with a needle that contains sperm. So this is how in vitro fertilization works. You add sperm to the egg to fertilize it and let the embryo develop before implanting it into a woman to develop fully. And so imagine if you just added those CRISPR molecules we were talking about into the mix with the sperm, you could potentially edit this egg cell because it has it has all the DNA it needs in that one cell, and edit it at that stage before it develops and divides a lot of times into the full embryo. And so that's what people have pursued in a small number of cases, and it is possible to do. So why is this so different from some of the other applications I'm talking about? Why is there so much debate? Well, there's a big difference between editing skin cells, blood cells, things that we call somatic cells. That's what I'm showing on the left. That's what we're editing in, Victoria Gray and in basically every other application, versus editing germ cells. So that means sperm and eggs and embryos. So the main differences here are that if you edit a somatic cell, these changes are not heritable. So they're only going to affect that one individual and they will not be passed on. Whereas with germ cells, if you edit those, these will be heritable. So that child, eventual child, and their descendants and offspring will be affected as well. So people sometimes say, we'd be potentially altering the course of human evolution. I think of it more as, if something does go wrong in a germ cell, that's going to be passed down. It's going to affect every cell of that person once they're grown, and it's going to be passed down as well. So I think of it personally a little bit more about the risk of doing such a dramatic change versus changing human evolution. But that's just my take. And so there's so many ethical questions surrounding this. Again, I don't have time to get into all of them, but I'll just give you a little summary. I'm sure there's many things coming to mind for you immediately as well. So as I said, there's technical risks. If we don't hit our target, that could be, there could be issues with cutting and making edits at the wrong place. If that happens, it's happening throughout the organism, or throughout the person. There's philosophical considerations. There are differing religious takes on manipulating human embryos to begin with, let alone, doing these sorts of newfangled editing techniques. I think I'm probably most concerned, besides the technical risks, with the social impacts. And I think this, honestly, all of these apply to other questions about genome editing too, but it's sort of particularly an issue in this case. So I'm putting money on the screen because I think there could be huge differentials in who's able to afford some of these treatments. So you could imagine if this is a really expensive procedure, maybe a very rich couple can go do IVF and add in this sort of enhancement or changes to their embryo, whereas other people might not be able to. And so because this is so far an expensive sort of approach, I think we could be risking furthering economic divides between people, especially when it comes to health. You don't want some people to be able to prevent an illness in an embryo before their kid is born, and other people to not be able to. And then finally, I put this Hulk picture here to represent enhancement. So there's nothing to say right now that if this were to be rolled out broadly, that people couldn't use it to not just prevent disease, but also to make certain enhancements to their future child. So you could, in theory, try to make a smarter kid or an aesthetically different looking kid. And I like to point out here that for the most part, we don't know how to do that. So as I said before, we don't know how every sequence of DNA is related to traits. There's some traits where we can't predict them well, especially something like intelligence or appearance. As an example, height is controlled by at least 700 different places in the genome. So we can't just say, oh, we'll edit the height gene and you'll have a tall child. You would have to do a much more complicated thing that we technically can't do right now. We don't have the technical ability. So a lot of the things people might do are kind of impossible with our current understanding, but I think it's important to think about what could be done in the future as our understanding of genetics improves. Okay, so this sort of raises this specter of designer babies. So designer babies just means babies that people are influencing in some way and changing their DNA makeup to make them have certain traits. And this actually did happen. So there have been three CRISPR edited humans born. This was a huge issue about 3 years ago now, that we called the CRISPR baby scandal, where these babies were born, and it was actually just like a horrific blunder. From a science perspective, it was sort of horrifying. It was one guy who did this, and he did a terrible job in how he did it, in addition to, I think, the world not really being ready for the technology to be used in this way quite yet. There had been a lot of discussion where I think 99% of the scientific community was saying, we should not do this. This is, the technology's not ready. People aren't aware of this. There's so many social implications we have to think through. And then this guy just kind of went forward anyway. And so I'm not going to go into too much detail on that, but I liked this. I liked 14 at least of the most damning details in this article. I didn't agree with all of them, but I think that this was a really good summary from Ed Yong, who kind of explained why it went so haywire and what really went down. So that was a big deal a couple of years ago. I mean, it's still a big deal, but I think that the sort of silver lining of this is that it's really reinvigorated discussions about how to make decisions on what we should let people do with genome editing and how we should regulate and oversee those efforts. And so I'll just call out two such efforts here. The first, I think this was one of the early important reports on this topic. There have been others, but this one from the National Academies of Science, Engineering, and Medicine came out in 2017, and it sort of outlined why that we're not ready from a technical perspective to move forward with germline editing in humans, and a lot of other interesting conclusions about when we could be ready or how that might look. But then the CRISPR babies thing still happened, even though this report was out there. And the guy who made those or edited those babies cited this and thought that this was sort of a green light for what he did. And so that means this wasn't the best report, or there's some ambiguity there. And so there's been more recent efforts to assemble additional committees and bodies to kind of come out with reports and come up with ideas for how to regulate and oversee this. And then the one example that I'll give is this report that came out from the World Health Organization just this year. So they assembled this expert advisory committee on developing global standards for governance and oversight of genome editing, and it's mostly germline editing in my mind, but it was broader. And so they came up with this document that's called a framework for governance. So they're starting to talk about how different countries might be able to kind of control this process, and how they might engage with people to inform those decisions. So this is very difficult. I think maybe Michelle can talk about what it's like to be on some of these committees and try to come up with these rules that can apply to the entire world. I think we're on the right track, but we could end up with a world where different countries handle this differently, which I think could lead to medical tourism where people travel to other countries to get procedures done. I'm also a little bit fearful that there will be just totally illegal clinics that pop up to do this, similar to how there are stem cell clinics that offer unapproved, terrible potential stem cell treatments. So I'm a little bit worried about that kind of thing, but I think for the most part, most scientists are not working on human germline editing, and I'm hopeful that it doesn't become a super predominant practice. My take on it. It's also, I think, totally fine to think that that's something we should be doing. It's really up to individual people to decide what they think of it. Okay, and so I like to kind of summarize here that, even though some of these things can be a little bit scary, there really are amazing things that we can be doing with technologies like this as well. And this is what we try to focus on at our institute. And so we can do really fun things in basic research to understand how nature works, to understand the diversity of life. There's amazing applications in medicine that are just emerging, like we discussed. And in agriculture, we can potentially combat climate change directly, or at least to make crops that can withstand the effects of climate change. There's all sorts of other things we can do there as well. So there's a lot that can be done. I think the technologies are advancing so much that we're becoming more and more limited simply by our own understanding and our own lack of creativity. So there's almost any kind of change you want to make in DNA at this point can be made, almost. So there's I think there'll be a lot of exciting science happening in the future because of this. Okay. And then I always just kind of like to recommend some further reading and resources for those that are really interested. My boss, Jennifer, wrote this book, A Crack in Creation, which is now a few years old, but I still think a really nice job introducing the topic. There's a newer book that's not on this slide called The Code Breaker, written by Walter Isaacson, that's also about the CRISPR field, and it's a little bit more up to date. And then I really like this human nature documentary that focuses on the discovery of CRISPR, the scientists behind it, what the process of science is like, and then all the things you can potentially do with CRISPR. And they have really interesting interviews with, a boy who has sickle cell disease who says, I don't think I would want this treatment because sickle cell has made me who I am. And a girl who has albinism, and her parents who say, we probably would have used germline editing to prevent this disease in our child, but now that we have her, we never would have done that because, again, this is part of what makes her who she is. So I won't explain the whole movie, but it's really interesting, and I think they have some nice insights into the ethical implications and what that might look like in the real world. And then our website is innovativegenomics.org. We're on various social media platforms if you want to kind of stay up to date on some CRISPR news and what we're doing. If you want to follow me on Twitter, I'm at the CRISPRist. I really only tweet about like very nerdy CRISPR biology stuff. So it's probably not the best follow. The IGI is a better one. Okay, so thanks so much for your attention, and I'd be happy to take any questions.
So Megan, thank you for this wonderful presentation. You've done it so clearly, and I just love that video of sort of visualizing the DNA strand and the CRISPR-Cas coming in and cutting it. It's just amazing to be able to think of it in that way. And you've taken us now the whole breadth of the technology to the applications. And thank you for so carefully, sort of saying what can be done now, what can't be done, the fact that the technology is still developing. And, there's still so much to be done. So I have a few questions for you, but I think I'd like to start off with the question that we have from Kelvin, because I think it's an excellent question. So he's asking, can CRISPR potentially be used to correct Down syndrome? And if so, at what stage of development can that be done? So I think that's a good one to start with.
Yeah, great question. I think potentially, yes. So CRISPR can potentially be used in almost any way. How realistic that is harder to say. I think when you're talking about genetic disorders that are caused by really large scale changes or many different changes in DNA, those are a lot more difficult to change. And so Down syndrome involves an extra copy of a chromosome, and that's huge, right? That's like a huge portion of the genome. And there have been studies where people have been trying to use CRISPR to remove entire chromosomes or entire stretches of chromosomes. So in theory, I think it could be done. I bet it could be done in an animal model. I'm not sure. I would think right now that the technology is quite safe enough to do that. I think making such a huge change, if it goes a little bit wrong, if you targeted the wrong chromosome or something, or you remove part of the chromosome, it might not be clear what kind of effect that would have. So I would be a little bit dubious about what amount of information I would want to see and experimental data and everything before testing that in being. So that's the technical perspective. So I think it is possible, maybe a little bit more difficult than treating other disorders. I think on the other hand, on the sort of social, ethical, moral side of things, I think a lot of people with Down syndrome have wonderful lives and, don't necessarily want or need to have their disorder corrected. I think that's a very personal decision. It's different for every family. And so, yeah, I think that would be a difficult decision to make. And then in terms of the when in development, that was the other part of the question that would need to happen. I think that it would be difficult to do that in an adult. I think that would be something that would be more of a candidate for germline editing, if that were to become a technology that is approved and used and everything. Just because if you have developmental delays and the brain develops a particular way, a lot of that stuff is sort of locked once it happens. So once you develop and organs and tissues and everything are the way they are, changing the DNA underlying that might not make a huge change to that person's life. So the reason sickle cell is one of the furthest along and why that can be curative is because you're making new red blood cells all the time. And so if you made that correction and all the red blood cells that you made became healthy, the disease would be gone. But if you're trying to do something like edit the brain, or edit some, the bones and muscles and everything, it's a little bit more difficult because they're either not dividing cells that aren't getting turned over, or they might just be really slow to divide. So yeah, Down syndrome is an interesting one to think about because it does have all these different considerations.
Yeah, thank you so much, Megan. I think hugely complicated because, as you say, it involves usually the entire chromosome. So at this stage, we're really targeting with the technology very small mutations, usually single bases. So yeah, so probably a bit of a way off there. So Megan, I think I'm going to take Stephen's two questions together because one, he's asking, how is the CRISPR RNA delivered to a specific spot on the DNA strand? And then if you change one piece of DNA, what can it potentially do to the rest of the genome of the organism or the DNA of the organism?
Yeah, great questions. So I didn't get into how we deliver any of the CRISPR machinery. And because it's really complicated and it varies by what you're trying to do. So if I were trying to edit, like the butterflies that I mentioned a few times, in that case, people inject the CRISPR machinery directly into the embryo of the butterfly. So as it's developing, they can do these little micro injections. And so that's like a very clear physical process. And that can be done like in the human germline, like I said, with IVF. But for the most part, people are not doing that for research. And for most medical applications, you can't just like directly inject CRISPR into somebody. So I guess I would say for things where we're working with cells in the lab in a dish, we can do a lot of different things. So one of the prominent ways is to sort of electrocute the cells. So you give them a little electrical jolt and zap them, and the DNA, CRISPR machinery, and however you're delivering it, all gets into the cells that way. You can also use a virus to deliver the machinery. So that's what's kind of the most prominent strategy so far in medical applications, is that you have a virus sort of package all of that machinery and then deliver it into cells. So viruses have evolved basically, right, to inject their genomes into cells. And so we just co-opt viruses and take out all the things that make them nasty and keep in just the CRISPR parts. Or put in CRISPR parts, they're not there originally. So there's all different strategies. It really does depend on the application. But this is also a limitation of clinical applications. So there are a lot of diseases that I would say we basically can't treat right now because we have no good way of getting these editing machineries into those cells. I think the brain is a good example of that. So there's some conditions like sickle cell, we can take out stem cells, treat them in the lab, put them back in, and those make all the new blood cells. A brain we can't take out, treat in the lab, and put back in. And so we have to come up with ways of getting these components to those organs and tissues. And it's just very difficult. And it's an area where there has been significant investment, I would say, recently from people that fund research to kind of drive new ideas forward, because we do need new ones.
And Megan, could you comment on the specificity? Yes.
Yes. Yes. So I think there's kind of two questions in that one. I'll just answer both to make sure I'm covering it. So one question is, I think you said, how does that change in the DNA get to all the other cells? If you put CRISPR into one cell, how is it going to all of them? That's not quite how it works. You actually have to have that edit happen in every single cell that you want to change. Or you have to edit stem cells that then make new cells. So you'd have to, like if we were editing for cystic fibrosis, you'd want that CRISPR protein to get into every single lung cell and make that correction of the mutation in every single one. And then the other question is, the other interpretation, I would say, the specificity of the enzyme. So if it cuts at one site in the genome, how do you know it's not going to cut somewhere else as well? And does it do that? So I would say for the most part, like 99% of the time, it cuts where it's supposed to, and we have edits just at the target site. And then maybe 1% of the time or less, it totally depends on the experiment, the application, but 1%-ish of the time or even lower, you'll see an edit somewhere else. So sometimes, since we're programming this thing with this 20 nucleotide stretch of RNA, sometimes you might have maybe 18 of those letters of the RNA match somewhere else in the genome. And that kind of tricks Cas9 into landing there and cutting there. That's called an off-target edit. And so that can happen, and it's something you have to think about when you're designing your guide RNA. And there's all these tools online you can look at if you want to see what it looks like to design good guide RNAs that only cut where you want. Honestly, they're kind of bad at predicting this. So the way that most people do it is they'll just try a whole bunch of different guide RNAs and see if I'm just targeting one gene, I can probably cut multiple places to still knock it out. And so they'll test multiple and see, okay, does this one have bad off targets? Is this one? So you kind of have to compare empirically. And I think the other thing that we haven't met here that I'm more concerned about, I think, for medical applications is not off targets, because I think we kind of have them under control for the most part, but on target changes that aren't the changes we want. So sometimes if Cas9 cuts and say we want to, fix the sickle cell mutation, you might cut at that mutation, try to paste in some piece of DNA that's healthy, but instead the cell deletes like a huge chunk. So instead of reacting the way you want it to, the cell's repair proteins just go and do whatever they wanted to do and cut out a big portion. I think that does happen also a very small percentage of the time, but those types of changes could have really big impacts, potentially in causing cancers or just killing the cell outright. And so that's something that I think people are monitoring in clinical trials, and people are becoming increasingly aware of it happening, despite sort of missing issue for a long time. So again, I don't think that's a deal breaker for these therapies, but it's important to be aware of it and be looking for it to make sure it's not causing issues.
Right, because I think the technology is developing quite fast, that people are, looking at new things like base editing and, sort of getting the CRISPR-Cas to do things in a slightly different way so that it's more specific. But,
Yep.
I was interested when you started your talk and you spoke about 23andMe and the different mutations for cystic fibrosis. Now, we're in Africa, and we know that the most common African mutation is also single base change. But often people don't look for it because they're not expecting it. And you did hint a little bit at, sort of causing health inequity through this technology, because it's sort of aimed primarily at wealthy European origin populations. I don't know if you want to comment a little bit on how we can think about making it more accessible to those who need it most, because when you use the example of sickle cell, of course, Africa is the place where it occurs most, with about 5 million people affected, India with about 1 million people affected. So how can we think about it being applied there?
Yeah, absolutely. I mean, I could probably give a whole talk only on that topic. There's so much to cover. Yeah, I mean, this is something we talk about like daily at our institute at this point. It's a huge focus for us, and I think I'm a little bit hopeful that because people are so aware of it, it'll mean there's some kind of progress, but who knows? Yeah, it's a huge concern, especially, so we are developing a sickle cell therapy, and we do want it to extend to anyone who'd like to access it. And I think right now we're on track for this therapy and similar ones to cost $1 million a treatment, on average. It could be 2 million, it could be 500,000, whatever, but a lot of money. And it's not necessarily going to be covered by insurance. There are examples of other therapies like gene therapies that are not covered by certain people's plans, and then they're, mortgaging their house to try to get some treatment for their kid. And there's obvious inequities that are already kind of happening. And so we're talking about this a lot. I think it's going to take multiple approaches to make it happen, to make it accessible. So we're talking about technological solutions on one hand. So things like this delivery issue, for example, with sickle cell, right now we take cells out of patients, treat them in the lab, and put them back in. That requires all kinds of equipment, people handling cells in the lab. They have to have a certain level of expertise. I believe you need a clean room, like it's all kinds of expensive things. But if we were to develop an in vivo delivery mechanism, like a shot, meaning directly in the body, the edit is happening, that could circumvent a ton of those costs that go into developing the therapy. So we are trying to develop those sorts of in vivo delivery technologies to simplify the process. We're also trying to think about, different models of economic development or economic models for how to distribute therapies. That's something we're about to start doing. We're starting what we've been calling an affordability task force to assemble health economists and drug developers and IP people, intellectual property experts, all kinds of people to talk about what options we really have. Like, should we give this to a company and then the company has to make money back because they're serving their shareholders and they have to charge a certain amount? Can we keep it nonprofit? Are there paths forward that way? How can we potentially scale this to other continents? Like right now, I imagine we're only going to be treating 12 people in Oakland, California. That's not an entire continent. That's not all of India. And so we're thinking about, I don't think there are clear solutions right now. I think we need a lot of technological development and also social development and just healthcare access and how we roll out therapies.
Yeah, I think specifically pertinent in Africa, and if you don't have access to basic health care, those are the people most burdened by sickle cell. So if you could do germline editing for sickle in Africa, that would be extraordinary. But then we have to also think that you would need the whole assisted reproductive technology And that in itself requires, you know, the expertise of the clinicians, the laboratory scientists, and so on. So I'm hoping that when you're thinking about doing this, Megan, that you're also going to include some African clinicians and scientists on your panels, you know, to talk about things that may be more practicable in a setting like South Africa, like Africa generally. So I just want to
definitely.
Yeah, thank you. So I want to go to Charles's question about how far is the technology in unlocking the secret in aging and death? That's an interesting
man. Yeah. I would say don't listen to me about aging research. It's not really my forte. I honestly find it hard to follow. I think there are so many older billionaires that never want to die, and so they're funding all kinds of kind of like hype-filled companies and efforts devoted to anti-aging, and it can be hard to weed through and figure out what we really know and what's hype. I think we're learning things, and there's definitely progress about, you know, in understanding what causes aging and how it can be slowed, I guess. But I feel like we're still a long way off from really significantly extending people's lifespans or ever stopping something like death. I think it's a long way off personally, but there's some exciting things happening. I don't know, I'm very critical of it, or I have a hard time, or cynical. I don't totally believe we're going to get that far that quickly. I think most people have sort of shifted to talking about health span instead of lifespan. So trying to keep people healthier for a longer amount of time, preventing disease through all different sorts of ways, treating disease better, rather than trying to extend lifespan. But I don't know, maybe you have more insightful thoughts on that, Michelle.
So I think you're right. You know, it's very much like the example of intelligence, that we really don't know what contributes to it. And the same with aging and lifespan. It's probably not all genetic. There are probably other social factors, behavioral factors, and so on. So it will be a very complex question. And there's a lot of research trying to understand, you know, what is the genetic basis of healthy aging? But they haven't come up with very many genetic determinants for that. So I think I'm with you there, Megan, in terms of saying it's a long way off. There are lots of people working on it, but no sort of dramatic breakthroughs for sure. So so I just wanted to I've just put in the chat there, and I don't know if that can be somewhere that people can see it, a link to the International Commission that deliberated on what they call a clinical pathway to heritable human genome editing. And if somebody wants to understand a little bit more, there's a great executive summary there. And there will be a third international summit on human genome editing, which will take place in London in March next year. And so people who are interested can maybe look out for that. But I think we've come more or less to the end of our session. And Megan, I just want to thank you. And I emphasize also what Stephen is saying, excellent presentation, very articulate, very accessible. And I'm hoping that many will be watching this and learning from the presentation. I think you've given us a very grounded sort of sense of what it can be used for and where we're not yet there. And I think that's really important for people to understand that this is not yet the era of designer babies, for example, but that there is so much hope in terms of what can be done, and loving the example of sickle cell and watching that over the next 5 to 10 years to see whether that really comes to the clinic and really goes to different parts of the world that need it most. So Megan, our heartfelt thanks for this wonderful seminar. And in terms of SciFest Africa, this has been a really excellent session. Thank you so much.
Thanks so much for having me.